Okay, rounding out our microbiology session, we have Dr. Brittany Ross. Brittany did her PhD work at the University of Texas Medical Branch, and she's now a postdoc at the Georgia Institute of Technology, where she is funded by an F-32.
Thank you for that kind introduction. Thank you to everybody in my session for introducing how to kill bacteria and mechanisms of not to die in the bacteria. I am going to do slightly different, where bacteria don't die, but from broad mechanisms for every kind of killing. So for my postdoc, I'm in the Whiteley lab, which is a bacterial physiology lab, and they work mostly on Pseudomonas, and I work on the difficult pathogen Mycobacterium abscessus, or Mycobacterium abscessus. They just changed the name. And this is a model system of looking at physiology for chronic infecting bacteria. So first off, what is a chronic infecting bacteria? So unlike an acute infection resolves and the pathogen is completely cleared. Chronic infection is a hallmark with long-term colonization by a pathogen. So this may mean decades or years of colonization with no problems, no symptoms. The immune system wanes, bacteria grows, and now the patient has disease. So something like tuberculosis. Other organisms like Staph aureus, which is already introduced, initial infection usually has an acute infection with disease. However, you think you get better, but unbeknownst, there's still colonization by the pathogen. And this pathogen then comes back repeatedly every time the immune system wanes. So if you're really stressed out, you might get another skin colonization or skin infection for these organisms. Now, the problem with these long-term colonizations is they have high economic burden. So not only is treatment more costly and more common repeatedly, patients have reduced productivity because they're sick repeatedly. And this has a large economic burden, almost $1 billion in the US per year. And so, but what I'm interested in is the fact that these bacteria are present for long periods of time. That gives them more time to adapt to their environment, particularly the host environment. So there's many mechanisms of adaptation, and one may be that now they have more time to interact with co-infecting microbes, and they can have a higher chance of gaining resistance genes. And then maybe you'll need a treatment. Now your treatment doesn't work, so you're more likely to have death of the patient if you have a new resistance gene. But what I'm really interested in is the fact that these bacteria are constantly bombarded by the immune response and often multi-drug treatment regimens, but you can't get rid of them. They're just still there. And over time, these bacteria are exposed to these pressures, and that selects for populations that are better and better and better at surviving. So this phenomenon where these bacteria are not necessarily resistant, but surviving is known as bacterial tolerance or persistence. And so for my PhD, I was looking at these systems called toxidity toxin systems, which they sense the environment, and when they sense the stress they like or respond to, they alter the bacterial's physiology. They reduce the replication or transcription, and as a result, that makes the bacteria much more difficult to kill. So to kill a bacteria, they have to be replicating, or they have to be transcribing. If they're not really doing that, you can't really kill them. And that's even true for phage infection. You have to have a replicating bacteria. So, but for my PhD, the bacteria I was working with had over 100 of these systems, and they were not highly conserved. So trying to prevent the bacteria from sensing environment and changing its physiology to protect and promote a survival phenotype was not really a way to move forward to target these bacteria. So for my postdoc, I wanted to step back and ask a slightly naive question, and that is, what is the physiology of bacteria during chronic infection? Most of the data we know is based on in vitro data putting bacteria with antibiotics, not necessarily immune cells or all these different other stresses that they are exposed to. And so I wanted to take this approach for my postdoc, and with my main goal of being able to toggle bacterial physiology to promote clearance by the host or even just by treatment. And so that's why I started picking up this pathogen, Mycobacterium abscessus. There's three main reasons. When I joined the lab, there was 1,500 publications, so a good environment to start working on. It is an emerging pathogen globally. It's found in your sink and shower heads, and it is adapting to the human in real time. And so if you grow this bacteria on an agar plate, you find two things. So it can either look like a smooth colony morphology, which looks a lot like E. coli, or it produces this rough colony morphology, which is flat and flaky. And the general consensus is that the smooth colony is in the environment and causes initial infection. However, during infection, as time passes, what happens is patients have a point mutation in their bacterium, and that results in the transition to this rough colony morphology. And we know that the bacteria point mutation it is in the biosynthetic machinery of this glycopeptidolipid layer, which is situated up here at the surface of the cell wall. And so the loss of this changes the appearance of the bacteria to look like rough. And so with this transition, we also know that it has a problem for patients. It goes from being a more chronic infecting patient or chronic infecting disease to a more acute infecting disease. And we know this because patients with cystic fibrosis, this is a particular problem for them. These patients have a genetic disorder where they have increased mucus buildup in their airway, and so they get colonized by these chronic infecting bacteria, and it's almost impossible to get rid of. And so for these patients, they have lots of lung problems. Having MAB means that often they can't get lung transplants. And so what clinicians are doing is they're testing patients for this organism even before they have any symptoms or they think they have it. And then they just watch their progression over time. And what they found is that these smooth bacteria colonized patients have a more asymptomatic infection, so just chronic colonization. And then over time, the patients to have more and more problems. And when they have the colonization by rough bacteria, they have a lot more symptomology and a higher mortality. And what actually happens is these rough bacteria can make cavitary lesions just like tuberculosis. And so this pathogen is actually related to tuberculosis, and there has been some speculation that the evolution of this bacteria may be something what we think happened with tuberculosis to be a host-restricted, very successful pathogen. And so using this model system and with a single point mutation that's non-reversible, I can start asking questions like, what is the physiological differences here? Because that will tell me what is more important for a chronic infecting bacteria versus an acute infecting bacteria. And there's many ways to look at bacterial physiology. So you can look at metabolism or genes or transcriptomes. And what I'm focusing is on the top of the central dogma, the genes that are important for infection chronicity between the smooth and the rough bacterium. And so there has been some introductions to mutagenesis for bacteria. And for my experiments, I'm using mutant libraries, and we use transposons that jump into the genome, but they cannot jump out. And so if you give a transposon to a bacterial population, every single bacteria it jumps into a different region in the genome. And so I end up with these populations of heterogeneous mutants. And this allows for high throughput screening of genes that are important for survival in a particular environment. And so I have a mutant library for both the smooth and the rough, and about 40 to 50,000 mutants across the 4,900 genes. And before I put this in an animal to look at important genes for chronic infection, I just wanted to, sorry, it keeps going bored. I just wanted to ask, well, okay, what genes do they need in vitro? I would expect they would be pretty much the same in vitro. And that's mostly what I found. So 302 genes are needed for the smooth and the rough bacteria to survive in vitro, but there were also these genes that were only needed by one morphotype and not the other. And this is completely unheard of. You wouldn't think a single point mutation would change the genes the bacteria needs to survive. This would suggest that there's actually rewiring of the physiology by a single point mutation. And so to confirm these studies, I took a single gene from the rough bacterium, and this is just an example of one of them I tested, and I did CRISPR interference. And so when you turn CRISPR on, transcription turns off. And this bacterium, the smooth bacterium, this gene is not important for physiology. When I turn it on in the rough bacteria cannot. It grows a little bit, but then it stops and dies. And so I also tested this in clinical isolates from patients with cystic fibrosis. So these patients I'm monitoring as they transition in the clinic. And these two patients were originally colonized with a smooth bacteria. And you can see that their morphotype or smooth morphotypes have no physiologic effect when you have CRISPR. However, the rough bacteria that they now are colonized with, these genes are important for their rough morphotypes. So this suggests that the smooth and rough bacteria are physiologically distinct even before animal infections. And it gives me an idea of what the physiology is by looking at what genes are important in these two. And as often, things that are high regulatory remodeling genes like protein kinases. And so now I can use these libraries and ask what genes are important for infection chronicity. And to do this, I take my libraries, put them in a mouse, and ask what genes are no longer on the outside, no longer present after infection. And this is when I got some really exciting data that a majority of the genes the morphotypes need during infection are completely unique to the morphotype. There's very little overlap between the genes needed to survive in vivo. And so for the smooth bacteria, we see that there's genes that's important for resilience and adapting to pH changes. For the rough bacteria, we see that they're responding to host stresses and repairing their DNA. And so this tells us that the bacteria are physiologically distinct in the animal environment and during infection, and that they're likely interacting with the host different. And so from this data, I can conclude that simple morphotypic change, a single point mutation, can actually rewire bacteria, and that leads to different physiology, which has drastic ramifications because a lot of other bacteria change their morphotype. This is reproducible across clinical isolates, and that this allows us to understand the physiology of the bacteria in vivo using these models. As a result of this data, there's a lot of ramifications. So the bacteria, or these assays where you're looking at different mutations and identifying genes essential for survival, are often used to find new drug targets. However, if you use one isolate, you're not going to get a complete picture. You're thinking like, well, there's 302. Statistically, I would get it right. A gene that would be a good target. But that's not actually the case. So there's two genes that I've been working up in the lab, one that's unique to smooth and one that's unique to rough. And these are two genes that are actually being developed as new drug targets for tuberculosis with the hope of repurposing the drugs for M. abscessus. That's a problem because that would lead to selection or adaptation. So if you target the rough bacteria, you can capitalize on that. You can prevent the bacteria from transitioning to rough, buying patients more time with the asymptomatic smooth infection. However, if you target the smooth bacteria, now you're going to promote adaptation, and the patient's probably going to have a shorter lifespan. Additionally, there's this new data that we have that you don't traditionally find antibiotic targets by using these mutagenesis assays in an animal. And this data is important because it shows that across the morphotypes, not all the antibiotic targets would work effectively across them. But it also highlights a new opportunity. There's only 30 genes conserved that are important for both the smooth and the rough isolates to survive. But these genes, if you use them as drug targets, that they would only be good drug targets during infection. And this is important for in the realm of antimicrobial resistance, where resistance can evolve not only in the human infection, but in a sink drain to the hospital or in a lake from runoff. So if you have an antibiotic that only works during infection but never works outside, then you potentially have a chance of increasing the lifespan of an antibiotic because you can't ever select for resistance outside the human body. And so for my postdoc, this has mostly been work inside a test tube or in an animal model. But an animal model is not a human. And so I joined Marvin Whiteley's group simply because he was asking this great question. Well, what is the physiology of bacteria during human infection? And so in the lab, they get samples from cystic fibrosis patients, do RNA-seq on the sputum from these patients, and from that work, they've been looking at the transcriptomes of Pseudomonas aeruginosa and been able to ascertain the physiological footprint of bacteria infection in humans. And using that data, they then can ask, well, how good is the model that I'm using in lab? How good is LB? It's actually not that good. But then you can take this data and compare a whole bunch of models to the human data and improve your assay that you're using in the laboratory. So you're actually looking at physiology that represents true infection physiology. So doing this for MAB, it's a lot more complicated. There's really low abundance of mRNA for bacteria. MAB is a low abundant bacteria, so it's even more complicated for that. And so I'm using two methods to enrich for bacterial RNA. One using the fact that it's a high GC genome and the human is not. And this has increased my bacterial RNA for MAB from 30% of the genome to 80% of the genome that I have gene coverage for. And additionally, I'm using these probes that are specific for MAB to pull out RNA that is only MAB RNA, so I can enrich for those, not spend a whole bunch of money sequencing all the way through the sample. And so for my postdoc, I've been doing these broad omics studies to look at the physiology of bacteria during chronic infection, utilizing this chronic and acute morphotypes as my system. But there's a lot of unanswered questions. I have a lot of pathways that I'm going to follow up on, but there's also a question, well, what causes the transition? We have no idea. And they don't actually monitor transition in the clinic, so it's really hard to ascertain what is happening to the patient that causes the transition. And so for K22, I'm looking at what causes transition and seeing if I can use the genes that are important for the smooth or the rough to try to control the adaptation or prevent adaptation. Also, I want to get back to what's in the environment that promotes the survival phenotype. Because if we know what promotes the survival phenotype, the cues that initiate it, then we can try to stop that physiology from happening and disrupt the bacteria physiology enough that the host can deal with the clearance. And so my whole goal is being able to root my data and all my experiments in situ physiology data. So if a gene is not expressed in the human, I'm not going to work in the lab. And again, my whole goal is to be able to toggle bacterial physiology, either to promote clearance by the host immune system or treatment, but also potentially not try to clear the bacteria, prevent disease. Like you can keep the bacteria, but it's not causing problems, then that's not really a big deal, right? And with that, it's important to note that morphotypic change is actually quite a broad phenomenon. Most of these bacteria are on the WHO watch list or CDC watch list, so there's a room to expand my laboratory on multiple organisms, and many of these are important for chronic infection as well. So with that, I'd like to thank my wonderful lab, as well as my postdoc committee and my funding source, and the wonderful tech that I work in the lab, Emma, who's been very helpful with getting all this data.
All right, thanks. Who has questions? Oh.
Maeve from MSU. It was a really good talk. So I was wondering, so heterogeneity in a population is often at a transcriptional level, but do you know if you look at clinical isolates from an initial stage, if there are a subset that are already rough that are just selected for upon infection, or if it's an actual switch that they cause a point mutation rather than just a selection of a few that were already present in the population and then overtake?
So I'm monitoring patients over time, and what you'll see, and this is also consistent with the literature, is what you'll see is like the patient has smooth, there's no rough, then they'll be co-colonized if you capture it. It's very like quick, it seems like. Okay. They'll be co-colonized, and then they only have rough. So there is no information about how these two morphotypes interact. I feel like the rough bacteria is like, hey, something's going on, transition, and because there is no, like if I compete them, they're happy together. So I think it's promoting transmission of the smooth, but that's not necessarily known, but something I do want to work on in the future. And I also find that like if you co-culture them, they actually directly interact, which is weird because the rough bacteria is hydrophobic and the smooth bacteria is hydrophilic. So you would not expect that to happen.
Lindsay from Albert Einstein. So very nice talk. I had a question about now with cystic fibrosis and the CFTR modulator therapies that are quite effective.
Yeah.
Is there much known about the transitions in the bacteria in those patients that maybe have a reversion from some of the phenotype or the, mucus, heavy mucus?
So you can't ever revert from rough to smooth. At least it's not, no. I want to do a Griffith experiment to try it, but that is never shown. But the patients who are going on Trikafta, they actually have lower burden of MAB, it seems like, but you can't really say they're clearing it because this pathogen is really hard to even like test for in the first place. You have to decontaminate the sample before you even grow it in the clinical lab. So you're already killing a lot of bacteria. So Trikafta is helping patients that have MAB, but now I'm trying to set up collaboration with people who have bronchiostasis, where the Trikafta drug is not going to necessarily work. And these patients, a lot of them are starting to have MAB. So there's like clinicians that only have bronchiostasis patients that have MAB. So it's a whole other population to work with.
Did you see? Does somebody have their hand up?
I'm just looking.
Sorry, I'm just still hung up on your last answer. What do you mean about not disinfecting? What did you say? You get the patient sample and you have to like kill some of the bacteria before you study the other
bacteria? So this bacteria is a slow grower. So my boss is not a fan of it. So he's not going to work on it anymore after me. But you have to take the sputum sample and then you treat it with like all these harsh chemicals to get rid of fast growing bacteria. And MAB survives better because it's got a super thick cell wall. And so you take whatever's left and then you plate it, and that helps you from getting contamination from other organisms because you have to grow the plates for like 7 days to test for them out.
Okay. If there are no more questions, let's thank Brittany.